Good morning, students. Once again, I'm meeting you to discuss about the lesson digestion absorption. So, in the last class, we have discussed about digestion in the buccal cavity and digestion in the stomach. I given questions. Some questions answer is there in the book, and I want to discuss two questions what I asked in the assignment 5. Number 1. What would happen if bicarbonate's level is reduced in saliva? Now the saliva contains bicarbonate. Its pH is maintained by the electrolytes like bicarbonates. That pH is maintained because of the bicarbonates. That is about pH 5.4 to 7.4. This is the pH of saliva. It's ranging normally 6.8 average. But such a pH of saliva is maintained because of the bicarbonates found in saliva. Suppose actually the enzyme salivary amylase is working in alkaline pH and optimum pH, average 6.8. This is the range only 5.4 to 7.4, but the average pH of saliva 6.8. Suppose if the bicarbonates level in saliva is reduced, what would happen? The saliva becomes acidic. When the saliva becomes acidic, what would happen? The tooth enamel gets dissolved or eroded. The tooth enamel, the enamel is, you know that one the hardest secretion I mentioned, that enamel gets actually removed or dissolved in an acidic pH. That's why what would happen if bicarbonate's level in the saliva is reduced? The pH decreases, the saliva becomes acidic. What is the effect of acidic pH? It causes what we have normally, the enamel of the tooth get dissolved. This is answer 1 for one of the questions. And the question I asked, what would happen? What would happen if hydrochloric acid is not secreted in the stomach? For that you have to recall the functions of hydrochloric acid. Now hydrochloric acid is responsible for providing acidic medium. And the second one, it converts, that is what is called inactive pepsinogen to active peps. And also it prevents putrefaction, that is what is called anaerobic breakdown of protein. So 2 points I am giving, so many points, we will take only 2 points. If hydrochloric acid is not secreted, what will happen? The acidic pH normally not maintained in the stomach. So no activation of pepsinogen to pepsin. Only by hydrochloric acid, the pepsinogen gets converted to pepsin. If there is no pepsin, protein digestion does not take place. That is one of the effects if hydrochloric acid is absent. Another effect, putrefaction occurs in the stomach. That is nothing but anaerobic breakdown of protein. Putrefaction means anaerobic breakdown of protein that causes bad smell, release of various gases, etc. So, if hydrochloric acid is present, putrefaction is prevented. But if hydrochloric acid is absent, putrefaction occurs in stomach. So these are the two effects when hydrochloric acid is absent in the stomach. So these are the two questions I asked in the assignment. So note it down for your reference. Now so far I studied what we have the digestion in the buccal cavity, digestion in the stomach. Now we have to study what happens to the food in the small intestine. Otherwise we can say digestion in the small intestine. Now let's see the process of digestion in small intestine. See, normally the food which is partially digested in the stomach, what is called a chyme, the creamy liquid enters into the duodenum. So duodenum is the major part of digestion. It's not the place of absorption, it's concerned with what we call the digestion. So digestion in the small intestine means it refers to the digestion of food materials in the duodenum. Now in the duodenum normally 3 different types of secretions are released from 3 different types of glands. Number 1, pancreatic juice from pancreas. Number 2, bile from liver. This is one pancreatic juice from, that is pancreas. The next one what we have, bile from liver. The third one, intestinal juice. that is from the intestinal glands. So these are the three types of secretions released into the duodenum or the small intestine concerned with the digestion of different food materials. Already we have partially digested food that is released from the stomach into the duodenum, what is called the chyme. This is the partially digested food. Now, actually what is happening? We have some movements in the intestine too, peristaltic movements. So these movements generated by what is called the muscular layer of the small intestine that help in mixing the food thoroughly with various secretions of the intestine, thereby facilitating the digestion. So we have peristaltic movements generated by what we call the muscular layer of the small intestine, which help in thorough mixing of the food with various secretions of what we have the intestine, thereby facilitating the process of digestion. Now we have to study the role of pancreatic juice, the role of bile, the role of intestinal juice. So the digestion of small intestine is a 5 marks question. Write short notes on digestion of the small intestine is a 5 marks question. Okay, prepare that one. What are the things to be added starting from here till the end? You have to take it. Okay, now let's see what's the role of pancreatic juice from pancreas. Now, what is the role played by pancreatic juice in digestion? So, normally this fluid is alkaline in nature because it contains bicarbonates secreted by the pancreas having a pH of 7.8. We'll discuss later. Now, the pancreatic juice contains the following 6 enzymes. Number 1, trypsinogen. Any enzyme ending with the suffix ogen, this is an inactive form. If we have ogen, that refers to its inactive form. Zymogen, inactive form enzyme. So trypsinogen, inactive. Then chymotrypsinogen, inactive. Then carboxypeptidase, the third enzyme. These 3 enzymes are protein digesting enzymes, proteolytic enzymes. Now, pancreatic amylase. So we have salivary amylase. Now here pancreatic amylase. The role is the same as in the case of salivary amylase. Digesting starch and glycogen. Pancreatic lipids. It's the one concerned with the breakdown of fats. You'll see. Nucleases. So the enzyme name is always having the suffix ase. Substrate plus ase. Substrate plus ase. Substrate is nothing but a substance on which a particular enzyme is acting. For example, if you are taking maltose, this is the sugar. Now it is acted upon by the enzyme maltase, ASE. So the enzyme name is always ending with the suffix ASE. Maltose, the sugar, maltase, the enzyme. Just only one letter is differing, that is OA. So anyway, here it's called nucleases concerned with the breakdown of nucleic acid. Now on 2 marks question, what is enterokinase? I mentioned this trypsinogen is an inactive form. So it's being activated into active trypsin by an enzyme called enterokinase. Enteron means you have the intestine or digestive tract. Kinase means activate. Kinase means activate. So it is secreted by the intestinal mucosa. It is concerned with what is called the activation of trypsinogen into trypsin. So the inactive trypsinogen gets converted to active trypsin by the enzyme enterokinase. So what is enterokinase? 2 marks question. Trypsinogen is activated by enzyme enterokinase, secreted by intestinal mucosa, the epithelium, into active trypsin. So I represent the equation. You can also represent the form of equation. Okay, that's about the enterokinase. And once formed, the trypsin activates chymotrypsinogen. Chymotrypsinogen, that is activated into what is called chymotrypsin. So the activation of trypsinogen is by enterokinase, and the activation of chymotrypsinogen is by trypsin itself. Now let's see the role of each and every enzyme, how far they are working. Chymotrypsinogen, the enzyme in active form, is activated by the trypsin to form chymotrypsin. This is activation. Trypsin is actually activated from chymotrypsinogen with the help of enterokinase. Now this chymotrypsinogen gets converted into chymotrypsin by trypsin. Now I mentioned about there are three proteolytic enzymes. Let's see first what is the role played by these proteolytic enzymes. Now we have the proteins present in the food not digested and the partially digested proteins in the chyme. On reaching what is called the intestine are acted upon by the following proteolytic enzymes. Acted upon by the following proteolytic enzymes. Okay, now, so this is about action of proteolytic enzymes. We have different proteolytic enzymes. They are acting on what we have, the food. Now, what are the proteolytic enzymes? Number one, trypsin. This is number one proteolytic enzyme. Now, what is its role? It hydrolyzes proteins into the next products, peptones and polypeptides. I represented here, the proteins get converted into polypeptides and peptones by what is called trypsin, a proteolytic enzyme. Likewise, chymotrypsin is also responsible for the breakdown of proteins into polypeptides and peptones. But it breaks or hydrolyzes the peptide bonds associated with the specific amino acids. It doesn't break the peptide bonds between the amino acids of all. It breaks only specific amino acids. That's the bond between specific amino acids. For example, tyrosine, tryptophan and phenylalanine. That's the only difference between what we have trypsin and then chymotrypsin. So trypsin converts proteins into polypeptides and peptones, breaking the peptide bond at any place. But whereas the chymotrypsin, again it breaks the proteins into polypeptides and peptones, but it breaks the peptide bonds associated with the specific amino acids like tyrosine, tryptophan and phenylalanine. That's the difference, but the function would be the same. Both are breaking the proteins only, producing the next products. Chymotrypsin hydrolyzes the peptide bonds, not all the peptide bonds, what I mentioned earlier, associated with the specific amino acids. For example, between the tyrosine, tryptophan and phenylalanine. So bonds being broken only between specific amino acids. The next one, carboxypeptidase. We have classification carboxypeptidase and also just what we call aminopeptidase. I don't want to touch that one. Now it breaks the polypeptide chain into tripeptides and also amino acids, the resultant products, the further hydrolysis, the ultimate products. The next one, pancreatic amylase, is actually similar to the salivary amylase, digesting the starch into maltose. See, starch and glycogen, it's also another polysaccharide, both being acted upon by pancreatic amylase to maltose. Now, what is the difference between pancreatic, what is called amylase, and salivary amylase? Salivary amylase can digest only cooked starch, but this one, pancreatic amylase, digests both cooked starch and raw starch. Now, nucleases. Now, the nucleases are concerned with the breakdown of nucleic acid into nucleotides, the unit of nucleic acid, and also what we have the nucleosides. A nucleotide is formed of, you know that one, 3 units. 1, a pentose sugar, 2, a nitrogen base, and 3, a phosphoric acid or phosphate. Whereas a nucleoside is formed of only, you see that one, sugar and nitrogen base. Sugar and nitrogen base together called as nucleoside. When phosphate is added, it becomes nucleotide. We'll see the reaction, okay, in the intestinal juice. These enzymes also present in the intestinal juice to break down what we have the nucleotides and nucleosides into the respective components. Now the last enzyme in pancreatic juice is pancreatic lipids. It is a fat splitting enzyme. It acts on emulsified fat, the one which is being formed after breakdown by what we have the bile juice. I'll tell you what I mean by emulsification. So the emulsified fat is nothing but a triglyceride. What is a triglyceride? It is formed of 3 molecules of fatty acid plus 1 glycerol. So 3 fatty acids plus 1 glycerol together form what is called a triglyceride. What is a monoglyceride? It is formed of one fatty acid and one glycerol molecule. So I am using the terms here. So it acts on emulsified fat. So that what is happening, the fat is being broken into free fatty acid and then monoglycerides and sometimes diglycerides also. A diglyceride is formed of two fatty acids and one molecule of glycerol. So the monoglycerides are further actually broken or further hydrolyzed by the same enzyme, the lipase enzyme, into fatty acids and glycerol. I mentioned already monoglyceride formed of one fatty acid, one glycerol. Diglyceride is formed of two fatty acids and one glycerol. And triglyceride is formed of three fatty acids and one glycerol. That's all about what we call the enzymes of pancreatic juice. We have to go for the role of bile, a few words, a paragraph only. And afterwards the action of intestinal juice, only the reactions. Now let's see the role of bile in digestion. Bile is a fluid secreted by what we have, the liver. It contains two pigments. One is bilirubin, another one biliverdin. These are the two pigments present in the bile, responsible for the coloration of the bile and also for the coloration of the undigested waste. So it is nothing but the breakdown products of hemoglobin from the damaged and dead RBCs. The bile also contains bile salts, two salts, sodium glycocholate, sodium tauroglycocholate, two salts are present. The emulsification of fat is because of the bile salts only, not other components. So anyway we can say bile is responsible for emulsification. So it is performed by the bile salts, two bile salts. Then also contains cholesterol and phospholipids. These are the components. But the bile is a juice which doesn't contain any enzymes. No enzymes present in the bile. That's why it's concerned with only the emulsification, not the breakdown. What is the role? It's normally responsible for the emulsification of fat. The emulsification of fat. What is emulsification? So emulsion and emulsoid, these are the two types of colloids. What is a colloid? Colloid contains two components. One is a solute, another one is solvent. What is called dispersal phase and dispersal medium. There are two types of colloids. One is called emulsion, another one emulsoid. For example, oil and water is an example for emulsion. There are two phases. The solute and solvent do not mix with one another in emulsion. For example, here mentioned, just we see that one water, in which we have the oil droplets, they do not mix with what is called water. It is an example for emulsion. If you are taking a sugar solution, the sugar is completely dissolved in water. The solute and solvent mixed with one another. That is an example for emulsoid. Now we are taking large molecules of fats. The enzyme lipase cannot digest large molecules of fats. So that they should be broken into fat globules floating in water. Then they are acted upon by the lipase enzyme. That process is called emulsification. That is brought about by the bile salts. So bile salts are responsible for the emulsification of fats. So normally how it does? So the bile salts reduce the surface tension of fat droplets and break them down into fat globules. The fat globules are smaller than the fat droplets. And another function of what is called the bile, it also activates lipase, the enzyme, the only enzyme for the digestion of fat, to digest the lipids. Only enzyme, lipase, for the breakdown. We have different enzymes for digestion of carbohydrates and proteins, but for lipids, we have only one enzyme, but present in different places. Gastric lipase, small amount. Intestinal lipase, we have also digesting what we have the lipids. So lipase break down what is called lipids into fatty acids and glycerol, what is it already. That lipase is activated by what we have the bile. Okay, that's about the functions of bile. The last one here we have to go for the action of intestinal juice. Only just we have a few statements along with the reactions. the secretions of Brunner's glands, the one which is present in the submucous of duodenum, and other intestinal glands present in the wall of the intestine together constitute what we call intestinal juice or succus entericus, in Greek succus entericus. So the secretions of Brunner's glands and the intestinal glands together form the succus entericus. It contains a number of enzymes for splitting the disaccharides, for splitting the polypeptides, for splitting what we call the diglycerides and monoglycerides, to split nucleotides and nucleosides, etc. Now it contains the enzymes maltase, sucrase, lactase. These three are called disaccharadases, digesting disaccharides like maltose, sucrose and lactose. Then peptidases digest the di and tripeptides. Nucleotidases, they digest the nucleotides, a unit of nucleic acid. Now the nucleotide is normally formed of three units I mentioned above. One, sugar, a pentose sugar, a nitrogen base and what is called a phosphoric acid molecule. All present in nucleotides. When it is broken, it forms a nucleosides which contains only the nitrogenous base and sugar. That is being broken by the nucleosidases. So nucleotidases and nucleosidases. Nucleases breaking the nucleic acid into nucleotides or nucleosides. The nucleotides and nucleosides are broken further by nucleotidases and nucleosidases. Along with the lipases, here also it is called the lipase only. Is there any left lipids? In the form of diglycerides and monoglycerides, they are being broken into fatty acids and glycerol. Now here only equations represent the both. So maltase is acting on the malt sugar, the maltose found in the germinating cereals, maltose, sugar OAC, enzyme AAC. Now it is a disaccharide formed of two glucose molecules, glucose plus glucose. Then lactose, it is a milk sugar, acted upon with the lactase enzyme. It contains glucose and galactose, the monosaccharides, so we are getting what we call the two units of monosaccharides. Sucrose, normally it is the one found in cane sugar. It is acted upon by sucrase enzyme to form one molecule of glucose and one molecule of fructose. So we have all actually the sugars disaccharides, they are being broken into two monomeric units, the monosaccharides. The dipeptides and tripeptides, actually they are formed of two amino acids or three amino acids, they are being broken by peptidases. They may be called as dipeptidase or tripeptidase, releasing the amino acids, the last unit, the ultimate unit of protein. Okay, that's about 4. And we have to know the functions of nucleotides, nucleotidases, and lipases. Nucleotides, the unit of nucleic acid, are hydrolyzed by nucleotidase enzymes so as to form nucleosides and phosphoric acid. Now the nucleosides are further hydrolyzed by nucleosidases to form sugar and nitrogen base. That's why I mentioned normally a nucleotide is formed of sugar, nitrogen base and phosphoric acid. Now the diglycerides and monoglycerides, which are formed because of the digestion, are acted upon with the lipids of the intestinal juice, so that we are getting the units, ultimate simpler units, fatty acids and glycerol. Now the mucus along with bicarbonate ions of the pancreas or the pancreatic juice provide an alkaline medium in the form of pH 7.8 for the various enzymes to act on the different materials. For the enzymatic action, the alkaline pH is provided by the bicarbonate ions as well as what with the mucus of the pancreatic juice. Okay, so what is the ultimate product? We'll see. As a result of digestion, we have received the simpler units. Okay, that is the end of what is called the digestion of the small intestine. And right on the board, I will tell you something about it. The result of digestion. As a result of digestion, the complex macromolecules of food like carbohydrates, proteins and lipids are converted into their respective or corresponding monomeric units, simpler units. Monomeric or simpler units, because the complex food, the macromolecules are polymers. They are being converted into monomeric units. For example, the carbohydrates are converted into the monosaccharides, the simple sugars. We have three types of simple sugars: hexoses, glucose, fructose and galactose. Then the proteins are converted into the unit amino acids. There are nearly 20 different amino acids in each. Now the lipids, the fatty materials and the derivatives together called as the lipids. The lipids are broken down into fatty acids and glycerol. These are the ultimate end products. So these end products are entering into the intestine. They are being absorbed in the ileum and jejunum region. What will happen about the absorption of these materials we will discuss in the next class. Thank you.